Good day, this is the Professor, CBMD PhD, and today is Friday, June 16, 2023. It is 5:11 and 7 seconds, a.m. Eastern Daylight Time, and today's research, brain tumors. So let's hop to it here. Open Analysis Platform for Pediatric Brain Tumors provides robust data resource for childhood cancer research. Date May 31st, 2023. Source: Children's Hospital of Philadelphia. With the help of thousands of genomically sequenced samples, researchers have used this platform to identify initial findings about genetic variants associated with poor outcomes that could help guide future diagnostic and therapeutic advances. As pediatric brain tumors are collectively the leading cause of cancer-related death in children in the United States. However, the severity of pediatric brain tumors varies wildly, with some having an almost universally fatal prognosis, while others have relatively strong long-term survival rates. Though all brain tumors negatively impact these children and their families to at least some degree. The limited access to tissue samples and patient-derived cell lines has been a significant barrier to understanding the differences between pediatric brain tumors at a molecular level. That long sought-after data could lead to better diagnostic techniques and potential targeted therapies that can treat these deadly tumors. In 2011, the scientific researchers began extracting and preparing what has now become nearly 6,000 tumor samples, with over 68,000 subsamples. More than 1,000 of these tumors were sequenced to form the initial release of the Pediatric Brain Tumor Atlas in 2018. The research is accessible to anyone conducting research who is looking for new therapeutic targets or finding new ways to translate research into clinical practice. At the time of the study, the research contained genomic and clinical data for more than 1,000 pediatric brain tumors and 22 patient-derived cell lines. The research provides an open, real-time framework to genomically characterize pediatric brain tumors. It is the first large-scale, collaborative, open analysis of genomic data that provides a cloud-based resource for researchers looking for more comprehensive data on pediatric brain tumors. As collaboration is key to accelerating new cure discovery, research made it possible for experts across the globe to come together and gain a deeper understanding of the leading cause of cancer-related death in children and young adults. The facility is already providing researchers with more insight into potential drivers of pediatric brain tumors. In this study, researchers found that the loss of the tumor suppressor gene, TP53, is a significant marker for poor overall survival in fast-growing brain and spinal cord tumors called ependymyomas, a certain diffuse midline gliomas, and dysregulation of the gene was also implicated in hypermutant high-grade gliomas. As solving pediatric brain tumors cannot be accomplished by any one institution, the facility model of shared real-time collaboration supported by researchers has not only empowered new discoveries, but also innovative ways of performing the required science on behalf of accelerated, collaborative innovation for children affected by brain tumors. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.